Sarah Eldridge, and I'm currently going for my Professional Science Masters in Applied Mathematics at UMass Lowell. Prior to this, I graduated from the University of Vermont with a degree in biology and spent 3 years working in a neurobiology lab, which focused on studying taste. Today I'm going to present to you research from the article A Conditioned Aversion Study of Sucrose and SC45647 Taste in TRPM5 Knockout Mice, conducted by Megan Eady, Benjamin Eschel, Darlene Peterson, Nathan Loris, Robert Margolski, and Eugene DeLay. The article was published in 2012 and can be found in the 37th volume of the journal Chemical Senses. The research presented in this study is very similar to what I spent my 3 years working on in the lab, although I did more genetic-based work while this is a behavioral study. Given the diverse background of this group, I'm sure many of you are unfamiliar with studies of this nature, so I'll begin with some general information on taste and the test subjects used in this experiment before diving into the research itself. There are 5 basic tastes: sweet, bitter, sour, salty, and umami, also described as savory. The ability to taste not only helps us enjoy food, but can also prevent us from ingesting potentially harmful substances. For example, we can detect that milk is spoiled almost instantaneously. Our bodies register that something is off and tell us not to drink it. Generally, in nature, a bitter taste is considered a warning that a substance may be poisonous or harmful. Understanding the mechanisms through which we perceive taste is fundamental to understanding how food choice may be influenced and can have implications for clinical populations with dietary challenges, such as diabetics or the elderly. One important phenomenon in taste research is taste aversion. Taste aversion can occur when we eat something and become sick shortly after. Sometimes the substance we ingested is not the cause of our sickness, but we still develop an aversion for the taste because our brain forms a connection between that substance and our sickness. For example, you may come down with a stomach bug after eating perfectly good salmon, and although the salmon is not what's making you sick, you may subconsciously link the taste to your sickness and avoid eating salmon in the future. In this study, and many other behavioral taste studies, mice are conditioned to have a taste aversion to a particular substance by injecting them with lithium chloride to create a nauseous feeling after they ingest the desired substance. This creates a link between the taste of the substance and the nauseous, thereby forming a taste aversion in the mice. Different tastes have different transduction pathways. This study focuses on the TRPM5 channel, which is short for Transient Receptor Potential Cation Channel of the M or melastatin subfamily member, number 5. There are a total of 8 different TRPM channels with different functions in the body. In particular, the TRPM5 channel is a key component in the transduction of bitter, sweet, and umami tastes. Salty and sour tastes are transduced through different receptors. Essentially, these channels activate when we ingest one of these tastes and sends a signal in the form of an action potential to our brains to let us know what we are tasting. Assumably, without this TRPM5 channel, you would not be able to detect bitter, sweet, or umami taste, and this is where the knockout mice come in handy. Knockout mice are genetically engineered so that an existing gene is replaced by an artificial piece of DNA to change the phenotype of the mouse, whether it be appearance, behavior, or a biochemical characteristic. Currently, mice are the genetically closest animal to humans, which the knockout technique can be easily performed on, and millions of knockout mice are used in genetic studies every year for this reason. In general, labs will engineer a few knockout mice, then breed them, rather than engineering each mouse used in studies. In this experiment, TRPM5 knockout mice are used, and they are missing the TRPM5 channel. Previous studies using the TRPM5 knockout mice focused mainly on the bitter taste, and found that knockout mice with a conditioned taste aversion to bitter substances only slightly avoided them. This suggests that the TRPM5 channel plays an important role in bitter taste transduction, but there may be another pathway as well which signals bitter taste. This study focuses on the sweet taste, and whether or not the TRPM5 channels are solely responsible for signaling a sweet taste, or if, like bitter taste, there may exist other pathways which can signal sweetness as well. To assess the role of the TRPM5 channel in sweet taste transduction, sucrose, a natural sweetener, and SC45647, an artificial sweetener, were used. All testing took place in a specially designed computer-controlled liquimeter. The apparatus is a clear chamber with one stainless steel wall that has an opening for the sipper tubes of bottles. A shutter covers the opening until the computer opens it for a set amount of time, allowing access to the drink. Multiple bottles sit in a tray, which the computer moves laterally across the apparatus, allowing access to only one drink at a time. The computer is also able to count the number of times a mouse contacts or licks each bottle, since the sipper tubes are equipped with sensors. 32 mice were tested, 16 knockout and 16 wild type for control. The mice were equally divided into a sucrose test group and an SC45647 test group. Prior to experimentation, the mice were put on a 22-hour per day water deprivation schedule so they would drink when testing occurred. This schedule was approved by an Animal Care and Use Committee. For the first 5 days of the experiment, the mice were trained to lick water from the apparatus. On day 6, the mice were presented with their conditioned stimulus, either sucrose or SC45647, as well as 33 other solutions. Immediately following exposure to the conditioned stimulus, the mice were randomly selected to receive a lithium chloride injection to induce nausea or a saline injection for control. Day 7 was a rest day, and the procedure was repeated the following day, followed by 2 more days of recovery. On day 11, the mice were tested with 6 solutions consisting of water, 0.05 and 0.5 millimolar SC45647, 30 and 30 millimolar sucrose, and 75 millimolar sodium chloride, which served as a control to ensure the mice were actually avoiding the sweet stimulus and not just any taste stimulus. Day 12 was another rest day, and the 6 solutions were presented again on day 13 to the mice. In general, the experiment found that the wild-type mice treated with lithium chloride injections developed a strong aversion for their conditioned stimulus, which generalized to the stimulus they did not receive, meaning that if the mice were conditioned to avoid sucrose, they also avoided SC45647 when presented with it, and vice versa. This was based on a comparison of the lick rates with the saline-injected wild-type mice who did not avoid either substance. The knockout mice exhibited an aversion as well, however it was much weaker than that of the wild-type mice. To ensure any lack of avoidance of the sweet taste by the knockout mice was due to a change in taste signaling and not an inability to associate the lithium chloride-induced nausea with the conditioned substance, a brief conditioned flavor aversion test was performed. This test associated a banana-like odor with sweet substances. It was found that the knockout mice did develop an aversion with the odor present, suggesting that the lack of avoidance exhibited by the mice in the taste test was due to a change in taste signaling. Overall, the study suggests that the TRPM5 knockout mice are capable of detecting sucrose and SC45647, but the sensory signal is compromised by the genetic deletion of the TRPM5 channels. However, based on the conditioned flavor aversion test, it should not be ruled out that those knockout mice who did avoid the sweet substances after the conditioning period may have detected the sucrose and SC45647 through olfactory cues rather than taste cues. However, this cannot be confirmed or denied by this experiment, as odor was not a factor in the conditioning. In summary, the results of this experiment suggest the TRPM5 knockout mice exhibit a residual ability to detect sucrose and SC45647, suggesting that, like bitter taste, there may be another pathway for sweet signaling, and that there may be much we still don't know about how taste is transduced. I'd like to thank you for choosing to listen to my presentation. If you have any questions, please feel free to comment in the thread and I will do my best to get back to you.